The story began not with an insect or a bird, but with a plant. Deep in the Cretaceous period, on the ancient supercontinent Gondwana, the first milkweeds pushed through the soil. Their leaves oozed a sticky white sap when torn, a sap that contained a potent chemical defense. This milky fluid was laced with cardiac glycosides, compounds that would prove devastating to any creature that tried to digest it. For millions of years, this sap formed an almost impenetrable barrier. Herbivores that sampled the tough, fibrous leaves would find their mouths coated in the bitter latex. The toxins within targeted a fundamental cellular mechanism, the sodium-potassium pump. By binding to these pumps in heart muscle cells, the glycosides could disrupt the electrical signals governing the heartbeat, leading to weakness, arrhythmia, and often death. So the milkweed thrived, its chemical arsenal making it largely inedible. Its success was written in the empty spaces around it, a quiet zone where few browsers dared to venture. The plant's defense was absolute, a perfect toxic fortress. It was an evolutionary masterpiece that would, much later, set the stage for a far more complex drama. The milkweed's chemical fortress stood unbreached for eons, but evolution never rests. Millions of years later, as grasslands unfurled across North America, a new challenge emerged. A particular lineage of insects, ancestors of what we now call the large milkweed bug, began to creep across those same toxic leaves. Their mandibles snipped through the plant's tissues, and the sticky white sap welled up, coating their mouths. Inside the insect's gut, a silent biochemical revolution was underway. Where the cardiac glycosides should have seized and paralyzed, specialized enzymes now intercepted them. These molecular shears evolved over generations, snipped apart the toxin's complex structure. The poison that felled other herbivores was dismantled into harmless components before it could reach the bug's vital systems. The meal was digested, the nutrients absorbed, and the insect crawled on unharmed. This was more than mere tolerance. It was a fundamental rewriting of the rules. The large milkweed bug and its kin could now feast freely where nothing else dared. They multiplied, their populations becoming a constant hungry presence on the plants. The milkweed's ultimate defense had been cracked, not by brute force, but by a precise internal alchemy that turned a deadly meal into mere fuel. But the insect's tolerance evolved into something far more strategic. Instead of simply breaking down the cardiac glycosides, their bodies began to actively harvest them. Specialized cells lining their gut absorbed the potent molecules, pulling them from the digested plant matter directly into their circulatory system. The poison was no longer a threat to be neutralized. It had become a resource to be collected. The toxins circulated through the insect's hemolymph, its insect blood, and were deposited with deliberate precision. They concentrated in the outer layers of the exoskeleton and saturated the fluid within. This wasn't a passive accumulation. It was an active sequestration, a biological stockpiling that transformed the insect's entire body into a walking repository of the milkweed's chemical defense. This internal arsenal manifested on the outside. The large milkweed bug's vivid orange and black patterning, once perhaps just camouflage, now blazed with new meaning. The bold stripes became an honest advertisement, a visual signal written in poison. The insect had not just conquered the plant's defense, it had stolen the very weapon and now wore it as a uniform. The meadow's morning sun dappled through the oak leaves, catching the flash of a young blue jay's wings as it landed on a milkweed stalk. Its bright, curious eyes fixed on a slow-moving insect below, a large milkweed bug, its orange and black exoskeleton gleaming like a warning beacon. The bird cocked its head, then struck with a sharp, decisive peck. The bug's armored shell cracked under the jay's beak, releasing a surge of bitter, viscous fluid, the insect's hemolymph, now laced with the sequestered cardiac glycosides. The bird recoiled, shaking its head violently as the foul taste flooded its mouth. It tried to spit out the offending liquid, but the damage was already done. The toxins had begun their swift absorption through the delicate membranes of its throat and tongue. Within moments, a profound bitterness coated the jay's entire beak, a sensory alarm screaming that this meal was a terrible mistake. It fluttered back to a branch, feathers ruffled in distress as the first waves of nausea tightened its stomach. The insect's chemical defense, borrowed from the milkweed and stored for this exact moment, had just initiated a brutal lesson in survival. The blue jay's beak had barely closed around the insect when its body convulsed. A violent, shuddering retch seized its throat, and the bird disgorged the bitter mouthful in a spray of orange hemolymph and half-digested seeds. Its crop emptied in spasms, leaving the creature trembling on the branch, its vibrant feathers now matted and foul. Inside the bird's chest, a silent sabotage was underway. The ingested cardiac glycosides had entered its bloodstream and were binding to the sodium-potassium pumps embedded in the membranes of its heart muscle cells. These molecular pumps, essential for maintaining the electrical rhythm of each heartbeat, began to falter. The orderly flow of ions across the cell walls grew erratic. The disruption sent confusing signals through the bird's cardiac nerve network. Its heart, instead of beating in its steady, powerful thrum, began to stutter and skip. This arrhythmia triggered waves of profound nausea from its nervous system, a desperate physiological alarm. Weak and disoriented, the blue jay could only hunch against the bark, its world reduced to the sickening turmoil within its own body. The blue jay's convulsions eventually subsided. It remained hunched on the branch for hours, its feathers ruffled, breathing shallow and laboured. The poison had not killed it, but the experience had seared itself into the bird's memory. A deep, visceral association was forming, linking the wrenching sickness to the vivid pattern it had just consumed. Days later, fully recovered, the jay was foraging again, its head cocked as it spotted another flash of orange and black stripes on a milkweed leaf. Its body tensed. A primal alarm flared, not from instinct but from that single brutal lesson. Without hesitation, it turned and flew to a different part of the meadow, leaving the bug untouched. The aversion became permanent. For the rest of its life, that specific combination of colours triggered an immediate, automatic rejection. This was not a forgotten mistake, but a hardwired behavioural shield. The bird had learned to read the insect's warning advertisement, transforming a moment of agony into a lifetime of caution. That single sickening experience in the meadow didn't just protect one blue jay. It protected the entire local population of large milkweed bugs. The educated bird, now wary of the orange and black pattern, carried its hard-won knowledge back to its territory. It began to patrol the milkweed patches, not as a hunter, but as a living, breathing warning signal for its own kind. When the bird's fledglings begged for food, it demonstrated the lesson. A parent would pluck a milkweed bug from a stem, hold the struggling insect in its beak for the young to see, then toss it aside with a sharp, dismissive flick of its head. No words were spoken, but the message was unmistakable. This colourful morsel was not food, but poison. Through these repeated, silent rejections, a local culture of avoidance took root. An entire generation of young birds learned the warning without ever needing to taste the toxin themselves. This transferred knowledge became a powerful shield, allowing the chemically defended insects to thrive with a safety net woven not from silk, but from shared memory and instinct. The milkweed bug's successful defense created an opportunity that other insects could not ignore. Across the same meadows, A different, completely harmless beetle watched the blue jays recoil from the orange and black stripes. It bore no toxins, but its drab-brown coloration offered little protection. Over generations, a subtle shift began in its population. A few offspring emerged with brighter, warning-like patterns. These new, colourful beetles were palatable, but they looked the part. A bird that had learned its lesson with the milkweed bug would see the familiar stripes and veer away, its memory of sickness overriding curiosity. The mimic gained a shield without ever needing to eat a milkweed plant or manufacture a single drop of poison. Survival became a matter of costume. This deception, known as Batesian mimicry, sparked an evolutionary arms race. As the harmless mimics flourished, the reliability of the warning signal eroded. Predators might encounter more fakes than real toxic bugs, risking a taste test. This pressure forced the true milkweed bugs to refine their colours or increase their toxin load, while the mimics faced constant pressure to perfect their disguise. A delicate high-stakes game of visual bluff. To confirm what was happening in the meadow, scientists brought the phenomenon indoors. In sterile laboratory rooms, they placed milkweed bugs in clear plastic containers, each with a fresh sprig of the toxic plant. The insects fed voraciously, their mouthparts piercing the leaves and stems, drawing up the milky sap that would kill most other creatures. After several days of this controlled diet, researchers carefully collected the bugs. Wearing gloves, they dissected the insects under microscopes, separating the tough exoskeleton from the internal hemolymph. Each sample was then placed into a vial, its contents destined for a precise analytical machine called a mass spectrometer. The spectrometer's readout provided definitive proof. It mapped a chemical fingerprint identical to the milkweed's own cardiac glycosides, now concentrated within the bug's body. The data revealed the exact quantity, sometimes milligrams per gram of insect tissue, transforming a biological mystery into a measurable toxic fact. Back in the meadow, the milkweed plant stood as the silent architect of this entire chemical defense system. Its milky sap, rich with cardiac glycosides, flowed through every leaf and stem. This wasn't just a passive poison, it was the foundational currency of a complex economy of survival, moving from plant to insect and up the food chain, governing who ate and who was eaten. The toxin's journey didn't end with the vomiting bird. That single sickening experience rippled outward. The educated blue jay began scouring the meadow, its sharp eyes now trained to recognize and reject every flash of orange and black. It wasn't just avoiding one bug, it was rejecting an entire chemical class of prey, reshaping its hunting strategy around the memory of that poison. Thus, a simple plant compound orchestrated a vast, visible network of checks and balances. From the molecular pumps in a bird's heart cells to the learned behaviors passed between generations, the milkweed's poison dictated patterns across the ecosystem. It painted the insects in warning colors, trained the predators, and in doing so wove stability into the very fabric of the grassland community.